Hello, welcome to the next A-level biology lesson from the conscientious biologist, Ben Gallagher. This is the second one from the protein synthesis unit. There's 3 lessons in this. It's really important that you don't just suddenly start on this one, okay? If you already think you fully understand protein synthesis and transcription and translation, fine. But otherwise, please make sure you've watched protein synthesis 1 and ideally protein biochemistry before that, and really you need to have watched DNA biochemistry to have a good idea of everything that this video talks about. They're all available on my YouTube channel, so please do go and have a look. If you do have a look and you are using these, please do think about subscribing to my channel. And remember, I do release lots of extra information on both Instagram and Facebook. Thank you. So let's start off looking at something called the central dogma. Now, a dogma is kind of the core belief of any system. So all religions have kind of a dogma that they adhere to. The central dogma when it comes to biology is basically this process of protein synthesis that we've been talking about. That life is based on DNA, that DNA can be transcribed by transcription into mRNA, a kind of disposable copy, and that's done to make a copy of the coding strand, because remember it's the coding strand in DNA that has the code, and you do that by complementary base pairing with the template strand. So if you've got the template strand and you've got a C, if you put a G on there, that G should be the same as the G that was on the coding strand. So you make a copy. Remember though, mRNA contains uracil and not thymine, but we'll talk about differences between DNA and RNA in the next video, okay? What you've then got though is after transcription, you've got translation to make the polypeptide, and after you've got the polypeptide, there's protein folding to make the final protein with its final shape to have its final function. Now, the key thing here is that in translation, the ribosome reads the 3-base codon, so 3 bases long, and matches it with the anticodon of a tRNA molecule, which brings a specific amino acid. That's the key thing. That you've got this specific code that matches to a specific code on a tRNA, bring a specific amino acid to build a specific amino acid sequence, because it's that sequence in the polypeptide over there that controls how it folds up because of the interactions between the R groups. We covered that in the protein biochemistry lesson 2 lessons back, okay? Now, the really important thing for you to understand is how that's kept so specific during translation. How can you make sure that the amino acids that you're getting match up to the DNA code and that your sequence of amino acids is always correct and specific? Well, the way that you do that is same as you would translate any kind of language. You have some kind of device, a code that can translate from one to the other. That's what we're looking at in this next slide. So you have what's called the universal code, and there's lots of different diagrams to show this, but they all look kind of similar to this, where you look at the bases that you have in your codon. Remember, the mRNA is read in those 3 base long codons, and those codons will always match up to the amino acids shown on this diagram. There's lots of different ways to have this diagram, so you just have to see whatever they give you in the exam and interpret it. But on this one, you look at your codon and you go, well, what's the nitrogenous base is in the first position, and you look down in that column. What's the second base? What's the third base? So for example, if you had CUG, you'd go, right, well, C is the first position, U is the second position, G read right across, that's going to be leucine, okay, LEU, okay? Not that hard to understand this, okay? Simple tables that translate the codon into the amino acid. Now, all forms of life that we know of, everything that we've ever discovered, all adhere to this exact translation code. Bacteria, fungi, plants, all different animals, everything follows this. So if you've got CAG in everything would give you GLN, okay? That's the amino acid that corresponds to CAG, and it is in all forms of life. Now, because it's all forms of life have this, we name this universal. This code is universal. All life that we know about in the universe, and there are likely to be lots of other life, But all life on Earth certainly uses this code, this sequence of codons to give you those amino acids. So you know everything is interlinked. It's one of the most powerful bits of evidence we have for evolution. That why would everything have it unless there was that kind of common ancestor way back in the distant past? So DNA code is universal. You'll also notice there's generally more than one codon per amino acid. Now that helps minimize the impact of mutations, because if there's a change in one of the bases, you might still get the same amino acid given. If you look over on the far side, you can see that LEU, leucine, has got 6 codons. This leucine, LEU, shows up 6 times way over there. So you could have UUG or UUA or CUU, CUUC, CUA or CUG. They're all going to give, all those codons will still give you leucine. So it could be a bit of mutation and you'd still get the same one. Whereas if you look at TRP, closer to us up here, TRP, there's only one codon that would give you TRP, that's tryptophan, okay? That's UGG. Nothing else would give you tryptophan. So if you want a tryptophan, you've got to make absolutely certain that there's no mutations, that you've got UGG as your base codon there, okay? Now, because there's generally more than one codon per amino acid, the code's referred to as degenerate. It's not that this codon will always generate this amino acid. It can be that one or that one or that one will generate it. So it's not that that generates that, it's degenerate because there's many. The only one that it is specifically generate for is the tryptophan that we mentioned. So the DNA code is universal and degenerate, okay? Now what that means is whilst one amino acid may be coded for by several codons, one codon will only ever have one specific amino acid. So what I mean by that, the leucine over there, that amino acid may be coded for by several codons, but CUU will only ever code for leucine. So the codons are specific to the amino acid, but the amino acid may have multiple codons. That's key stuff to remember, but we're going to look at how to use this now, and we're going to apply it to a DNA strand. So if you're going to do this for yourself, which you will have to do in the exam, what I'm going to do, I'm going to put up a short piece of DNA on here. It's the same one we've used in previous videos, okay? I've got the coding strand, I've got the template strand. If we think about what happens in transcription, the strands separate from each other. You can build your mRNA strand. Now you absolutely need to be able to do that. It's just basic complementary base pairing and remembering that in mRNA you have the uracil instead of the thymine. Guaranteed marks if you can match up an mRNA to a DNA molecule. And always remember you've got to match it up to the template strand so that the mRNA look is identical in code to the coding strand. The only difference being the uracil where there's a thymine. Now after transcription, that mRNA will move out of the nucleus through the nuclear pores in the nuclear envelope. The DNA will close back up and that mRNA strand is now in the cytoplasm, where it will associate with a ribosome, probably on a rough endoplasmic reticulum. So that was transcription. The translation part now, we need our little translation device, this table. And we look at the first codon, which I've marked in red, and you see it's GCU. So we know the first position was G, so it's going to be inside this red box because that's the G that starts here. We know the next one's C, so it's going to be down there, isolating it to that middle box. Now, if you look at it, because the code's degenerate, it doesn't matter what the third base is, because everything in this box here now, look, will give me an alanine, ALA. But the third one was U, so it's that specific alanine. So that codon will give me, by translation, alanine amino acid as my first amino acid in my polypeptide. Now, because the codons are non-overlapping, meaning we've read that first one, GCU, we now jump right to the next 3. It doesn't shift down by 1 and you read the next. It's not GCU, then 1 down CUC, then 1 down UCA. It's those 3, then totally the next 3. They don't overlap, okay? CAG, if you use this above me, you should be able to work out, look, C on the first position, A on the second position, G on the next position. It's going to give you GLN, which is, can't remember, glycine. Then you can read the next one because they're non-overlapping. You can work out what that one is from the sequence above me. It's going to be valine. The last one, GCU, was the same as the first one. So that's going to be another alanine. Go back through and check that for yourself, because that's going to give you the polypeptide. That's the process of translation. So that's how you end up with your polypeptide, by using this that we have worked out as humans, as scientists. We've worked out which ones go with what. So what this now means is you can read DNA. You speak the language of DNA, as long as you've got this to help you translate the language. So just last point, to put some of the key words together from this presentation. You can refer to the DNA code as being universal, because every single organism uses this same code up here. It's degenerate, because there's more than one codon that can generate an amino acid. And it's non-overlapping, because you read one codon at a time and they don't overlap. So make sure you know those three key terms. Make sure you can do this for yourself. If you want to practice, dead easy, just write out a string of letters for yourself. Just A, G, C, T, write them out, big string in any order, and go, right, that's my coding strand. Same thing, if that's my coding strand, can I now build a template strand? Write that on to complementary base pair. Then go, right, well, from the template strand, can I make an mRNA strand? Do that, remembering the uracils instead of thymines. Once you've done that, you can go, well, can I now read this a codon at a time, use this table, and see if you can make your own polypeptides. Practice until you can absolutely do it, because this will be in your exam. So hopefully that wasn't too tricky, and most questions that you're going to get on protein synthesis will be more from the previous video, all the details of transcription and translation and using all the correct enzymes and the sequences and how everything works. But you will be asked to apply your knowledge by doing exactly what I've shown you in this video. So from here, I want you to head to this video. This is Protein Synthesis 2. This fills in the last details to complete your full understanding of how proteins are made and how the DNA and RNA relate to one another. So we look at introns and exons, post-transcriptional modification, then finally a comparison of DNA and RNA. There should be a link to this appearing just over there. Please do give this a like if you find it useful and helpful. Subscribe to my channel if you haven't done so already, and do consider having a look at my Facebook and Instagram channels and following me on there. Thank you. Good luck.